and commitments. We have three sciences planned to organize a webinar to provide a platform for the exchange of knowledge in areas of biological sciences. So this webinar is a 2 days program, 7th and 8th August, and there will be total 5 lectures by the eminent speakers. I am very much happy to mention here that we have received total 640 registrations from different colleges and universities from length and breadth of the country, from the places like Cochin, Trivandrum, Pondicherry, Kerala, Nagaland, Rajasthan, Uttarakhand, Jammu Kashmir, Chandigarh have joined us today on YouTube live session. To begin our inaugural session, I would request Professor Kallol K. Ghosh, Chairman of the webinar, to give the welcome address. Over to you, sir.
Sir, please unmute yourself.
Okay, sorry. Very good morning and cordial welcome. Respected Professor K.L. Verma, I have received a message from him that he is busy with a program of MHRD, that's the National Conclave, and Prime Minister delivering a lecture at the same time. Respected speaker, very distinguished scientist, Professor Anil Prakash, Barkatullah University, Bhopal, a young scientist, eminent, I must say, Dr. Sourav Datta, IISER, Bhopal, Dr. Amit Tripathi, Department of Biotechnology, New Delhi, Dr. Zenu Jha, Indira Gandhi Krishi University, Raipur, Dr. Mariam Sarwat, Amity Pharmaceutical Institute, Noida, convener Dr. Binu Joshi, and 640 participants. I am very happy that large number of biologists have registered for this program, and they are attending various parts of our country. A large number of even PhD students as well as our MSc students, 5-year integrated students are also attending this very important. It's an honor for me to welcome you all in this very important webinar on advances in biological science, sponsored by the world is struggling for this outbreak of coronavirus. We are realizing this thing, this pandemic and other things. But still, our university, for the last, I think, 3 months, we are continuously organizing various webinars under this various platform. And I take this opportunity to place on record our gratitude to our Honorable Vice Chancellor, Professor K.C.L. Verma. He is our actually source of inspiration. And he always motivates us to do some type of the academic excellence and overall development of the university as well as the student fraternity. We deem it a great honor to have today actually two very eminent biologists, Professor Anil Prakash and Dr. Sourav Datta among us. We extend a very warm welcome to them. And I really have no words to express my deep sense of gratitude to Dr. Anil Prakash and Dr. Sourav Datta for accepting our invitation. And being a convener of this public outreach center of our university, I always appreciate such type of program. And we know you also agree with me that knowledge sharing is knowledge squared. And a number of scientists will be delivering, I think 5 scientists will be delivering to initiate and foster the fruitful exchange of ideas and advances on biological science. By training, I am a physical chemist. I don't know much about biology, but I know that biology is a very, very important subject. And I thank Dr. Zenu Jha, and she has chosen some very interesting topics. Very interesting topics. It will be useful for all the scientists. Like trace of plant productivity. Trace. We are also facing trace due to pandemic. Plants are also there are some trace. Then genome editing technology. Not only that, we have chosen some very important topic also, neurodegeneration disorder, and some haploid produced in rice, etc. I must tell my audience, my student, the participant, that you'll be happy to know that there are, I do not know much about this, there are 2 national institutes regarding the biotic trace and abiotic trace. And one of the institutes is available in Raipur, that is the National Institute of Biotic Trace. And they are doing excellent work, research work and other things. So really it will be, I hope that it will be a very, very interesting and important webinar. So I request all the participants to attend this exciting and enriching sessions of the webinar very seriously. Do attend very seriously. And we need your feedback also. We need your feedback. So I am confident that after attending this webinar, and you definitely will find new ideas, fresh energy for your future research. And they are very eminent scientists. You can interact with them. And thank you very much. Once again, I welcome you all in this. Thank you. Thank you, Dr. Zenu Jha.
Thank you, sir. So now, to begin the scientific section. Before we begin the scientific session, I would take an opportunity to introduce our first speaker, Dr. Sourav Datta, to our participants. Is Associate Professor in Department of Biological Sciences, Indian Institute of Science, Education and Research, Bhopal, IISER, Bhopal. He is PhD in Cell and Molecular Biology, University student, postdoctorate from University of Oxford, UK. Dr. Datta is working in the area of plant cell and developmental biology. He has guided many PhD students, and he has several research publications to his credit, with total 883 citations and H index of 10. Dr. Datta has also been recipient of several awards and fellowships. Distinguished Teacher Award for 2019-20 from IISER Bhopal, PHSS Foundation Young Scientist Medal, Innovative Young Biotechnologist Award by Department of Biotechnology, India, Ramalinga Swami Fellowship by Department of Biotechnology, India, EMBO Long-Term Fellowship for Postdoctoral Research, Marie Curie Intra-European Fellowship for Postdoctoral Studies, has also been selected as a member of DBT Task Force on Basic Plant Biology, Agriculture and Frontier Areas. So now, with these words, I humbly request Dr. Sourav Datta to please begin with his deliberations on how plants cope with environmental stress. Over to you, Dr. Datta.
Okay, I'll just start sharing my screen.
Yes, please.
Yeah, can you see my screen?
No, sir, it's not visible.
I think it is visible to public, sir. It is visible to public, sir.
Okay.
It is visible.
Okay.
All right. Visible.
Okay. Okay, so thanks a lot to the organizers and especially Professor Kallal Ghosh and Dr. Zenu Joshi for this kind invitation. It's a pleasure to talk to all of you about a little bit about what we are doing here in IISER Bhopal. So the topic of my presentation today is how plants cope with environmental stress. And what I'm going to specifically talk about today, I will just show you in a couple of minutes. I want to start with, if it was a live session, I probably would have started with a question to the audience, especially to the young students, that how many of you have seen the movie Shakuntala Devi? So, but because it's a webinar, probably people cannot raise hands and I cannot have an interaction as it would have been in a live seminar. But I just want to let you know that probably if you have seen the movie, there is a dialogue from Vidya Balan where she says that we are humans, we have legs, we are not trees, we can move around. This is the irony about plants. Okay, so they are rooted, they cannot move around. They have to cope with whatever changes the environment is posing, whatever stress they face, right from the place where they have started their life, they cannot move from there. So this is to study stress tolerance, to understand how to cope with stress, probably no better organism than plants, who are these sessile organisms probably provide us the best evidence of how to cope with different kinds of stress. Now, I will just go specifically to what I'm going to talk about today. So today I will tell you, I'm just modifying the title a little bit. So today what I'm going to tell you is how plants shield and mediate lockdown to protect themselves. So I am doing this because as all of us are going through this, this period of stress, this lockdown. So I just want to pose this question, that is this lockdown true only for humans, or do plants also face, undergo these kind of stresses where they protect themselves by shielding and maybe mediating some lockdown to protect themselves? So before I start the lecture, let me just give the background about the lab and about what we are doing. So we call ourselves the PCDB lab, Plant Cell and Developmental Biology lab. This is an image of the lab, students working in the lab. And in our work, we use a model plant, which is called Arabidopsis thaliana. Yeah, so this is like the guinea pig of the plant world, where you do most of your basic research in this plant, and then from the outcomes that you get from this plant, you try to translate those knowledge into crop plants and for the benefit of society. So it's easy to work with this plant because of various reasons, which I'm not going into the details. So here in this, using this plant, we are looking into two aspects. One is that how light regulates the plant growth and development. And the other is related with nutrient uptake, where we deal with how plants deal with low phosphate stress or with aluminum toxicity in the soil. So these are the two major areas of the lab. Because this is a webinar for the basic sciences students, so in today's lecture, I thought of talking about two stories from the lab, which are more of basic knowledge rather than the applied work. So today I'll tell you two stories about the light aspect of the work which was going on in the lab. The first story, it was actually a serendipitous finding in the lab by a very brilliant graduate student in the lab, Arpita Yadav. So I'll just tell you a brief anecdote about how this project started. So this happened, so as I was telling you, we are interested in light, how plant development is regulated by light. So this girl, Arpita, she was working with one of these genes in the light signaling pathway. And what we normally do is in this Arabidopsis, it is very easy to create mutants. So where you will have loss of function of a particular gene, and then try to see what happens if you have mutated that gene, what kind of phenotype do you get? So she was working with one of these genes, and she had overexpressed these gene in this Arabidopsis plant, which I just showed you before. What you're seeing in this image is a plate. So this Arabidopsis has really tiny seeds, which you can grow even on plates containing a media. and then later on, once they grow, you move them to the soil. So what you are seeing here on the slide is a plate on which there are two sides of the plate. On one side, these are all Arabidopsis seedlings, very young Arabidopsis seedlings, which have come out after the germination. And in this experiment, what she was doing was she was comparing her her transgenic lines where she had overexpressed a gene with the wild type, and she was putting them in different light conditions. And this particular plate was put in the UVB chamber. So she was checking what is the effect of UVB light if you change the expression of this particular gene. So she was supposed to take this plate out after a week, but somehow she forgot about the plate for 3 weeks, and she took it out after 3 weeks. And when she took it out, what she saw was what you see on this where I'm pointing, this side of the plate. This is a white place, almost completely that is what it look like. And on the other side, what you see is the sequence where overexpressing this particular gene was working, and they appeared to remain green even under UVB. So we initially, when she brought this plate to me, so I asked her to repeat the experiment, and after repeating it a couple of times, we always got the same result. So we became interested, thinking that does this gene have some property in providing some tolerance to UV rays in plants? So we asked this question: how plants protect themselves from UV radiation, and does this gene have any role to play in this? So before I go into the details of the work, let me give you a background. So this is the solar light spectrum, and you can see that it can be divided into the visible spectrum and the ultraviolet radiations and the infrared radiations. The ultraviolet radiation can further be divided into UV A, B, and C based on the wavelength. The UVC, which is the most energetic, fortunately is blocked by the ozone layer, which covers the Earth's surface. But part of UVB and and UVA, they can penetrate through the ozone layer and can reach the surface of the Earth. You must have, students probably know and have heard a lot about the harmful effects of UV rays on humans in terms of skin cancer, cataract. So similarly, the UV rays can also affect plant health and development. Although the studies in the plant field is not as much as we have done with humans and with animals. Now, how does it affect is actually totally based on the dose of UV. So if you look here, that UV light, if the dose is high, then it is actually a stress response. It damages biomolecules such as DNA and proteins. It generates reactive oxygen species. On the other hand, if the dose is low, then the UV light can actually promote plant development. Okay, it can have a positive response. So not everything about UV is bad. It totally depends on whether the dose is high or low. High doses are definitely not beneficial. But low doses, you see that they can have some good effects on plants, such as the production of phenolics, the accumulation of some secondary metabolites in medicinal plants, enhances the food quality. So these are the beneficial aspects of a low dose of UVB in plants. Now, what do we know about how this UV signal is perceived by plants, and what is the signaling that is going on inside the plant at the molecular level? So UVB radiation, it is perceived by a photoreceptor in plants called UVR8. So this is a dimer, and upon UVB radiation, it monomerizes, and then it binds to a light signaling factor called COP1. And from there, a light signal transduction cascade happens, and finally you have this transcription factor Hi5, which regulates many genes. Many of them are involved in UV protection and acclimation and repair of UV damage. Okay? So many of these genes are kind of sunscreen compounds in plants, which are regulated through this pathway to provide the plants protection against UV rays. In this pathway, you also have some negative regulators like RUP1, RUP2, and there's one more called BBX24, which is also an inhibitor of this pathway. So in our lab, we are interested in these BBX proteins. So let me just give you a background about what are these BBX proteins. So they are zinc finger transcription factors. So they contain this zinc ligating B-box domains in their structure. So in Arabidopsis, the plant with which we primarily work with, there are 32 B-box proteins, which are divided into 5 structural groups based on the domains which they contain. Okay? And these B boxes are cysteine and histidine rich domains. So these B boxes, there can be 2 B boxes or 1 in the different Arabidopsis proteins, which the BBX proteins, which we call, which contain these B box domains. Besides the B boxes, there are also this valine chlorine pair motif and the CCT domain, which are present in some of these B box proteins. So the gene which Arpita was working with was BBX31, which belongs to this group 5, and this is the gene which she has which when she overexpressed gave tolerance to plants against UVB. So before we went deeper into how this BBX31 is doing the job, we first went back to the literature to check, is there any indication which suggests that BBX31 might have a role to play in UV protection, UV signaling. So there was a paper back in 2004 in PNAS where there was a high throughput study, microarray study, where Hi5 was identified as a major player in the UV signaling pathway. And Hi5 being a transcription factor, all the genes which were regulated by Hi5 were identified in this microarray analysis. And if you look at that list, there was a long list of genes which are regulated. And in that list, under transcriptional regulation, the first gene which was identified was this Constance B box zinc finger family protein. And this gene turns out to be BBX31. So somehow in the last 16 years, no one really actually picked up this gene to find its role in UV regulation, but it was hiding in the literature that there might be some role of this gene. More recently, another paper from another very good big group, Gareth Jenkins group in Glasgow, they showed that this UVR8, which is the receptor for UV light, that acts as a chromatin modifier, and it can do histone modifications in several targets. And one of the target of this UVR8 is also BBX31. So there were evidences in the literature which suggested to us that we are on the right track. There must be something with this gene in playing a role in UV signaling pathway. So we asked the questions, what is the role of BBX31 in light signaling first? And second, how does BBX31 provide tolerance to UVB? So to start this work, the first thing that Arpita did was to check the expression of this gene BBX31. This codes for a very small protein, 121 amino acid protein. In its promoter, there is something called a UV box, which is a 4 nucleotide box, which are present often in genes which are regulated by UV. And what you see here is in gray, you see a response to white light in the expression of BBX31. And in black, you see the response to UVB light. So what you see is that the expression of BBX31, it increases, and around 1 hour after UVB treatment, it peaks. Okay, so it's a UVB induced. Specifically by UVB, this gene gets induced. We also checked how the expression is regulated by different doses of UV. So UV1, 2, and 3, they represent different like low to high dose of UV, and we see that when you give higher dose of UV, the expression of BBX31 increases. Next, we went on to see that I showed you the pathway, signaling pathway, where UVR8 is the receptor for UV signals, and then COP1 and Hi5 are major players in the UV signaling pathway. So we went on to check that if you mutate these genes, what happens to the expression of BBX31? So normally, col0 is the wild type. This is the control. Black bars indicate white light. Gray bars indicate expression under UV light. What you see is that the expression of BBX31 goes up under UV light. But if you mutate any of these genes, which are the major players in the UV signaling pathway, the expression comes down, which indicates that it is in this canonical UV signaling pathway, probably acting downstream of these factors. And the most downstream of them is Hi5, and which is a transcription factor. So probably BBX31 is acting downstream to 31. To check this, we looked at the promoter of BBX31, and we found that there is a 6 nucleotide element called G box, the nucleotide sequence CSC-GTG, which is a box where many of these light signaling factors are known to bind, including Hi5. So we wanted to check that is Hi5 binding to the promoter of BBX31 to drive its expression. We did an EMSA assay, and we found that indeed Hi5 binds to the G box. If you mutate the G box, you lose the binding. So we found that Hi5 is binding to the promoter of BBX31. We also did a chip assay followed by qPCR, and in this chip assay also we could see that there is enrichment of high 5 on the promoter of BBX31. But what we were quite intrigued was we found that this enrichment is happening both in minus UV as well as plus UV conditions. What that means, so this is that enrichment, this is the control, and this is where you see that there is enhanced binding of BBX of high 5 to the BBX31 promoter. So what this meant was high 5 binds to the BBX31 irrespective of whether there is UV or not UV. But when you look at the expression, then you see that this regulation of expression is very specific to UV conditions. So under UV, the expression goes up. So high 5 is constitutively binding to the 31 promoter, but enhances its transcript levels only under UVB. Now, next we went on to get some mutants of this gene to find out what role it plays in light signaling and in UV response. So I'll just take you, because I don't know, like in the audience, there might be people who are not from plant biology backgrounds. I'll just take you, explain to you what you're seeing here. So what you're seeing here are very young Arabidopsis seedlings. Okay, this part of the seedling, so this is basically the seed coat. The seed has germinated. You probably know about sprouting. After the sprouting, this is the embryonic stem, which is known as hypocotyl. These are the embryonic leaves, which are known as cotyledons, and this is the root part of the seedling. This is col 0 is the wild type or the control plant. Minus and plus indicates absence or presence of UVB. And MIP1A and BBX31-1, these are two alleles of the BBX31 mutant, one gene silencing line and one CRISPR mutant. So these two alleles were used for the loss of function mutant. And 35S BBX31 indicates the overexpressor lines. There were two lines, number 1 and number 2, which were used in this study. So first of all, I want you to look at only at the minus, like without UV seedlings. What you see is this is the wild type col 0 seedling. The mutants are shorter. The hypocotyl is shorter compared to the wild type. The overexpressors are taller. The hypocotyl is longer. What this indicates is, so light has a tendency to keep the hypocotyl small or short, because the seedling, once it is buried under the soil, it will try to grow tall to reach to the light. But once it reaches light, it won't invest energy in elongation of the hypocotyl. Rather, it will invest energy in greening, opening up the cotyledons, and doing the autotrophic functions of the plant. So with light, you have a reduction in the hypocotyl. So, but if you have mutated a gene and the mutant gives you shorter hypocotyl and the overexpressor gives you taller, then that indicates that it is a negative regulator of photomorphogenesis. Okay, so that is why we are calling that BBX31 is a negative regulator of white light photomorphogenesis. In under UV light, there is again the tendency that the hypocotyl length becomes shorter. So you see the wild type without UV and plus UV, the length of the hypocotyl is shorter, and that happens in all the seedlings. You will have shortening. But when you look at the percentage of reduction in the hypocotyl length, you see that it is much more in wild type compared to the mutants. Okay, so this would indicate that it is a positive regulator in the UVB signaling. So this shows that there is some differences in the way it acts in white light and in UVB light. Now, the high 5, which is a major player in the UV pathway, and I showed you that high 5 regulates, can bind to the promoter of BBX31 and regulate its expression. So we also made double mutants with this high 5 to check their interaction, the interaction between BBX31 and high 5. And what I want you to concentrate here, these seedlings I have already shown you in the previous figure. What you see is high 5 is a positive regulator of photomorphogenesis, meaning that it makes the mutant has tall hypocotyl, whereas 31 is a negative regulator, it has short hypocotyl. When we made a double mutant, the hypocotyl length was intermediate, which indicates that probably they are acting independent of each other. And furthermore, if you have an overexpressor of 31 which is tall, and if you overexpress this in the high 5 mutant background, you get this super tall seedlings, which again indicates that probably there is an additive effect between these two, leading to an even taller phenotype, and they are acting independent of each other. So this is the quantification of what has been shown in the image here. To further validate this, what we did was we did an RNA-seq analysis, where we looked at the genes which are regulated by Hi5 and 31. And after the transcriptome analysis was done, what we found was there was a very less overlap between the genes which were regulated by Hi5 and by 31. Okay, so this told us that that again this tells us that these two genes regulate the different groups of genes, and they are probably acting independent of each other to regulate the photomorphogenesis process. Now let's get back to the original question with which Arpita started the project, which was about UV protection. So here you see this is the wild type plants. If you mutate BBX31, they bleach more. But if you overexpress BBX31, these are three different lines. They remain green under UVB light, indicating that they are more tolerant against the UVB rays. Now, to understand what is the mechanism, how is this working, we looked at some of the pigments which are which can to tolerate different kinds of stress. In terms of chlorophyll, we saw some upregulation in the chlorophyll levels in the overexpressors. Although in the mutant we did not see a significant lowering in from the wild type, which is again a stress pigment. There we saw that the mutants had slightly lower levels compared to wild type, whereas the overexpressors had slightly higher levels. We sent our samples for metabolomic analysis to IIT Mandi, to Dr. Shyama Sagapalli, and analysis of phenolic compounds, formaric acid, gallic acid, hydroxybenzoic acid, and anil, which were upper expressors. And these phenolic compounds are those compounds which I was talking to you during the introduction, which give the plant this enhanced protection towards UVB rays. Okay, so here we got some clue about what are the metabolites which are enhanced in this 35S BBX31 lines, giving them more tolerance towards UVB rays. Now, next we ask the question that that Hi5, which is a major player in the UV signaling pathway, does that play a role in this BBX31 mediated stress tolerance? Okay, so for photomorphogenesis we found they were independent, but for stress tolerance we looked at the phenotypes again. Here the plate shows these different genotypes called 0, Hi5, the 35S BBX31, and the double mutant in white light, and here the plate shows under UV. So if you concentrate under the UV plate, this is wild type. The Hi5 is more susceptible to UV. The 35S BBX31 UV. But if you overexpress 31 in Hi5 mutant, again they become susceptible to UV, similar to Hi5, which indicates that overexpression of 31 is not enough. You also require the Hi5. So Hi5 is required for this BBX31 mediated tolerance towards UV rays. So if you lose Hi5, the plants again become susceptible to UV. So this is the chlorophyll content of these seedlings which was measured, and exactly what I explained to you just now. And then we looked at how are the genes which are involved in this UV signaling pathway, how are they regulated in these different lines. And what we could see, we looked at least 5 groups of genes. So ellipse genes, these are early light-induced proteins. CHS, CHI, these are enzymes in the anthocyanin biosynthetic pathway. UVR2 is a enzyme which is involved in DNA repair. When we looked at the expression of these genes, we see that in Hi5 they are downregulated. In the overexpressor of 31, they are upregulated. But if you overexpress 31 in Hi5, again the levels come down. So this fits well with the phenotype which we just saw. So that Hi5 is required for this stress tolerance phenotype of BBX31. Now, because Hi5 is required, that would mean that Hi5 is coming downstream to BBX31. But previously, I showed you that Hi5 can bind to the promoter of BBX31. So we wondered that if there is some kind of reciprocal regulation between these two genes, 31 and Hi5, and for that we checked the expression of Hi5 in the loss and gain of function mutants of BBX31. In white light, we could not see a significant difference, but under UV light, in the overexpressor of 31, we saw a 10-fold increase in the. What this would indicate is expression. When you overexpress 31, the expression levels of Hi5 are regulating the transcript levels of Hi5 was dependent then this is down. Okay, so even if you overexpress, if you muted UBR8, then the expression came down. So what, in a nutshell, what I tried to tell you was that we identify which plays an important role in light signaling, both in white light. In white light, it acts as a negative regulator of photomorphosis. Okay. It is acting independent of high-five to regulate. But more importantly, it acts as it plays a positive role in stress tolerance under UVB, and this dependent on high-five, where high-five is regulating 31, and 31 can reciprocally regulate the expression of high-five. So this was the work which she had done. How this for understanding how plants prevent themselves, protect themselves from UV rays. Now let's move to the second. Okay, so the take home message here is that sometimes forgetting about your experiment might become just have to, for students, you just have to keep your eyes on the open. You never know that something good might come out of something that you have about. Now let's move to the second story. This is the lockdown story. Much related to the current situation. The reason I call it the lockdown story is not only because that the public were living under lockdown, but more because this work means we have plant mediated lockdown. This is for people who are not from plant background, but everyone probably knows the word scouting. Morning you often get up in breakfast and get sprouts. So the seeds that are once they germinated, and once they germinated, then you have the sprouted seed, and then on this sprout, until the seed you have a seedling. When you have this stem on the bud, this is established by itself by growing slowly. So this happens when the other conditions are able to by but if there is a stress condition, then the plant has a mechanism to have a and I don't know under stress conditions, the germination condition and output. After that, also the past the science of the process generation condition that it is not going to
sorry to interrupt you, sir. There is some disturbance in sound from your end.
Yeah.
Yeah, it is now. It is clear now, sir.
It is clear. Okay. Sorry about that. Okay. So yeah, I was trying to say that if the conditions are not favorable, then there will be a developmental arrest, and the seedling will not go ahead with the further steps. Now, this developmental arrest, they are mediated by a hormone present in plants, which was discovered almost 80 years ago, known as abscisic acid, which happen in early development of plants. So this is a dry seed, and once there is enough water, then there will be imbibition, and the testa or the seed coat will rupture, and slowly the radicle will emerge. This is probably for students from your school days. You have heard about radicle and tumule. So these are the early steps in the germination process by this hormone called abscisic acid. After the seed has germinated, there are this post-germinative growth process where the hypocotyl emerges and finally the seedling establishes. And even in these steps, there is an inhibition mediated by this hormone abscisic acid. Now, abscisic acid is an endogenous hormone but there are other factors from the environment which influence this decision making, whether to go ahead or not. And one of the factors which primarily determines this is light. Okay, and for germination, we know very well that germination is light will inhibit this action of ABA to promote the germination process. For post-germinative growth, what is the role of light? There was a knowledge gap here. We don't exactly know what is the mechanism, what is light doing here, what is the mechanism, how does this work? So this knowledge gap, to cover this knowledge gap, we asked this question, that what is the effect of light on this ABA-mediated inhibition of post-germinative seedling development? Before I go into the details of ABA, let me just tell you briefly about how a seedling develops, whether it's when it sees light or is it in darkness. So this is the kind of development that you will see if a seedling germinates in dark. I was telling you that it elongates its hypogotyle, it becomes very tall. This is because it is trying to reach out for the light when it is buried under the soil. This part is known as the cotyledons, and it will keep its cotyledons closed and it will make an apical hook. The purpose of this is to prevent the meristem part from which the whole adult plant will develop. Okay, so this kind of development is called scotomorphogenic development in darkness. So long hypogotyle and closed folded cotyledons with apical meristem inside. Then the development is totally different. The hypogotyle becomes short, the cotyledons open up and they turn green in color. So here you see the development, short hypogotyle, the open cotyledons, and from here, from the meristem, the adult plant of the plant with true leaves and on the stem and everything will develop. So this kind of development which happens in light is called photomorphogenic development. Now, having shown that, now let us come to how is ABA regulating these initial steps in development. So what I showed you in this cartoon before was these are the original seedlings, how they look, and that was the schematic. So in darkness, you see this long hypogotyle closed yellow cotyledons, and in light, you see this short hypogotyle open green cotyledons. This is when there is no ABA present, 0 micromolar ABA. And this is in light and dark. Now, if you start increasing the concentration of ABA, then you see that ABA has an inhibitory effect on this development. So as you increase concentration, there is less and less of this development, post-germinative development happening. But if you notice carefully, you see that there is a stronger inhibition in darkness. Even in 0.5 micromolar ABA, you see that there is a very strong inhibition mediated by ABA. This is what has been shown here. So this is the 0 micromolar. So in black line, you have what happens in dark. In light, you have seedling establishment in light, in red line. So what you see is in light and darkness, there is a huge difference in seedling establishment rates in presence of 0.5 micromolar ABA or 1 micromolar ABA. So what I'm trying to say is that this inhibition is much stronger in darkness compared to in light. So this told us that probably there is some factor which is very active in darkness, which is mediating how this post-germinative development is happening. So we tried to understand what is this factor. And from the literature, there are many of these light factors which are more active in darkness. And one of them, a very prominent one, is COP1. So COP1, this gene, it was identified in a genetic screen back in the 90s. And the name COP1 was given, it stands for constitutively photomorphogenic 1. Okay, so you will find this figure in any plant physiology textbook. This is like the development which happens in wild type in dark and light, which I've already shown. This is the COP1 mutant. If you mutate COP1, then you see that in darkness it develops as if it is seeing light. It is not normal development in darkness. It has short hypogotyle, it has open cotyledons, although the greening will require some light. But overall, the morphology looks more like light grown seedlings. So that's why it is called constitutively photomorphogenic, as if this mutant is always seeing light. And this COP1, it is inhibited, it's a negative regulator of photomorphogenesis, and light inhibits COP1. Okay? So COP1 is one of those proteins which is more active in the darkness, but in light it gets inhibited. From the protein structure, you see that it has this ring domain, coil domain, and the WD40 domain. It's a 675 amino acid protein. And the COP1, the function of it is that it is a E3 ubiquitin ligase. So what it does is it ubiquitinates protein which are degraded through the 26S proteasome. And when you look at its accumulation, then you see that in dark it is accumulated in the nucleus, but in light there is a nucleocytoplasmic translocation of COP1, and thus the activity of COP1 reduces in light. So this we tried to look at what is COP1 doing in this post-germinative process and if it has any role. This is the wild type seedlings which I just showed you before, and this is what you get if you mutate COP1. So normally in darkness there is a very strong inhibition. In light also you see some inhibition, but if you mutate COP1, the inhibition is much lesser. Okay, so the loss of COP1 will the ABA mediated sensitivity of seedlings requires COP1. If you mutate COP1, then ABA cannot inhibit the post-germinative seedling growth. They will still germinate and start, still develop the cotyledons and start bringing. So these processes will continue both in light as well as in darkness. So this is what has been quantified here. So this indicates that the ABA-induced inhibition of post-germinative growth is COP1 dependent. Now, COP1 is a very ancient protein. Okay, and it's very ubiquitous. So it is not that only plants have COP1. Animals also have COP1. Humans have COP1. In humans, it plays a very important role in tumor suppression together with P53 genes. And if you look at the like the presence of COP1 in different plants, in ancient plants, you find them in almost all plant groups. So right from the transition from the water to the land, you find them in the mosses, in the ferns, gymnosperms, you find them everywhere. So we asked this question that whether the role of this COP1 is evolutionarily conserved. We did here was we took the COP1 from the moss Fiscomitrella patens and we overexpressed it in the COP1 mutant in Arabidopsis. What we could see was the COP1 mutant, which was hyposensitive to ABA. And now if you overexpress the Fiscomitrella, the moss COP1, it again becomes hypersensitive. The sensitivity increases towards ABA, indicating that the role of COP1 is evolutionarily conserved. So the moss COP1 can also complement the rescue, the Arabidopsis, the dicot COP1. And this is what has been quantified here. So it's a very evolutionarily conserved protein, and its role in this post-germinative growth is also quite evolutionarily conserved. Now, the next thing was to find out what is this COP1 doing in this ABA signaling pathway to regulate this post-germinative development. So here we went back into the literature and we got a PNAS paper from 2000, where ABI5, which is a major player in ABA signaling pathway, was shown to mediate this post-germinative developmental arrest checkpoint. So we wondered whether COP1 has any interaction with this ABI5 to modulate this developmental arrest or the lockdown which we were looking into. And here what we did was we overexpressed ABI5 in COP1 mutant background. And what we saw was when you do this, the phenotype of these lines are similar to COP1. So what this means is COP1 is acting downstream to ABI5. This is a quantification of the seedling establishment in these different genotypes. So basically, just to show you what we finally got was that that COP1 is acting downstream to ABI5. So this is the ABI5 in the ABA signaling pathway. So we wondered that what is it doing to the function of this ABI5, because at the transcriptional and translational level, we could not find any difference in ABI5 in the COP1 mutant. But what we could find was that in the COP1 mutant, the binding of ABI5 is a transcription factor. The binding of ABI5 to its direct target genes, this was affected in the COP1 mutant. So what you see here is the expression of these target genes, and the direct target genes in the COP1 mutant, their expression was lower. And also when we did a chip to check the binding, then we could find that in the COP1 mutant, which are shown in gray bars, there was less enrichment of the ABI5 on the promoters of these genes. So what I'm trying to tell you that we identified a factor called COP1, which is a major player in the light signaling pathway, but we found that it plays a role in the ABA signaling pathway. It acts downstream to ABI5 and prevents its binding to ABI target genes, which are ultimately mediating this arrest, the post-germinative growth arrest after germination. This work was done by another very talented graduate student, PhD student, Yadu Krishnan in the lab, and it got published in Plant Journal recently. And this paper, it got quite a lot of attention, and one of the senior editors from the journal, she thought that this work was very important, and she wrote a research highlight about this work in the same journal, where she named it "To Grow or Not to Grow." Okay, so it's like a very, very basic question that right at the beginning of the life cycle of the plant, how does it determine whether to go ahead or whether to arrest its growth, depending on the environmental conditions. This is why I was calling it a lockdown, that how the plants are mediating a lockdown when they assess that the growth conditions are not favorable. So why is this important? This is from a newspaper, Indian Express, 2nd August, so a couple of, like, not even a week back. You probably must have, some of you might have seen this news, that in Maharashtra, more than 1.5 lakh complaints, 83 FIRs against soybean seed germination failure. So seed germination and the next steps, the establishment of the seedling, this is very, very crucial, because everything related with yield, plant yield, everything depends on a successful germination and establishment of seedling. It all starts here. So something, if the seeds are not germinating well, the farmers really, really face a real problem. And this is a major issue with many kinds of plants, especially soybean and many other plants, where the germination and the post-germinative development cause major problems to the farmers. So that is why this, although the work is a very, very basic work, but it can have a lot of application in the field. So we are now looking into, so we, as I told you, we do the basic work in the lab, and then we try to translate the work into crop plants. So we have started some work in soybean, where we are looking into the function of this gene in crop plants. I'll just try to end with some of the other things which are going on in the lab, which I did not have time to talk about today. So as I told you that other than light, we are also interested in root development and how phosphate uptake for low phosphate tolerance, aluminum toxicity. These are some of the other areas in which my lab is working. And these are some of the publications from me and from my students in the recent years. Finally, I would like to acknowledge my team. So these are all my students in this t-shirts. This image was taken during the PhD viva of my first student who graduated, Arpita. So this is Arpita, who did the first work which I told you about UV tolerance. And this is Yadu Krishnan, who did the second work about this post-germinative growth arrest. And these are all the members in the lab. I would like to thank the funding from IISER Bhopal and from the funding agencies DST and DBT, which has helped in moving these projects forward. Also, I would like to thank my collaborators, Shyam Masakapalli at IIT Mandi, and Stephen and Hendrik. I would be happy to take some questions. So I just want to say that I know that there's a big student population in the which might be listening here. So the questions can be about the research work, or if you have any other questions related to your career prospects, about if you want to know something about IISER or any other questions, I would be happy to answer if I can.
Thank you. Thank you, Dr. Datta, for giving a deep insight of this plant's germination and environmental spread. And your talk was really very informative. And here I have received few questions from the participants that I will put here. The first question has been put by Sudhir Kumar Bhandari. His question is, why is Arabidopsis is the best model for genetics?
Yeah, so Arabidopsis, it like the life cycle of this plant. So it's like in 8 weeks. So it's from seed to seed, you can go in 8 weeks. So when you have to do genetics, when you have to create these double mutants, you have to look at several generations. Then you prefer to do it with a plant which has a shorter life cycle, which you can easily work in the lab. It is very easy to grow them in the lab, and also the genome is fully sequenced and very well annotated. So you have a lot of information already available. So all these things together with it can be transformed very easily. So this is a major factor. Many of the crop plants, they might have, the transformation might not be so easy. So all these factors add to the benefit of using Arabidopsis as a model plant.
Another question is about PR proteins. Like you are talking about the stress. So can you throw some light on PR proteins?
PR proteins are pathogenesis related proteins, so they are more involved in biotic stress. So what I talked primarily today was abiotic stress, about stress related to say drought. About germination, they can happen when there is drought or when there is the light is not optimum. So these are all abiotic factors which act as stress for plants. Together with this, there are also biotic factors which can act as stress. For example, pathogens. So during this kind of stress, when you are attacked by a pathogen, then these PR proteins come into action, and they have important role in biotic stress response.
There is one more question. What can be the best remedy for improving the germination?
This is like a very good question, but the answer is not so simple because there are several ways. As I was telling you that the germination requires all these optimum conditions in terms of light, water availability, proper humidity, temperature. So these are all factors which contribute, and they all have to be optimum for different kinds of plants. Different seeds have different optimum temperatures and optimum water requirement for this germination process to happen. What we are trying to do is to gain some insight at the molecular level, that what are the genes which are affecting these processes, so that in future we can probably modulate some of these genes to enhance that there is better germination. It is not only one. We cannot pinpoint that this is the most important. There are several things which are regulating this process.
Thank you, Dr. Datta, for this wonderful and well-explained talk. So now, moving further, I will take an opportunity to introduce our next speaker, Professor Anil Prakash. Prakash is presently working as Professor and Head of Microbiology at Barkatullah University, Bhopal. He has 33 years of teaching and research experience in the field of microbiology and biotechnology, specializing in mushroom, mycorrhizae, PGPR, and fungal biotechnology areas. Dean, Faculty of Life Sciences, and worked holding various administrative positions in the university. Professor Anil Prakash was conferred Fellow of Microbiological Society India and Fellow of Science, Engineering and Technology in contribution in the field of microbiology. He was also recipient of non-usual postdoctoral fellowship awarded by Government of India. Professor Amit Tripathi has published more than 60 papers in international journals and also contributed number of chapters in books published by Stranger Nature, CRC, and Elsevier. He has also been honored by the Department of Science and Technology by giving him Young Scientist Project on improvement of strain by protoplast fusion technology. He has also received two international projects sanctioned by Department of Biotechnology, Government of India, and has edited several books. So now I request Professor Anil Prakash to please begin with his today's talk on mitigating the impact of stress on plant productivity using plant growth promoting microbes. Over to you, Professor Prakash.
Okay, thank you very much. Uh, First of all, I would like to thank the director, Anil Bush, and Dr. Zenu Joshi for inviting me in this particular webinar. And our previous speaker, Dr. Sourav, has given you much insight on the stress, particularly on the basis of molecular biology. But today I am going to speak on this how the microbes are helpful in mitigating the stress, which is okay, slide shows. Okay, no problem. So just I would like to introduce about say about something about the stress. You know very well that previously speaker has told you about the stress, and we talked about more on the light stress. And the light stress, of course, play a very important role in the growth of the plant. And particularly, and there are some other biotic stresses is there, and abiotic stresses are also there, so that the growth of the plant affects. And there are number, and when we use, and we know that when we use the chemical fertilizers, that also harm. Everybody is now telling that if you are going to use the chemical fertilizer, the it will affect the, of course, 1 or 2 or 3 years, the growth will be very fast and give the productivity very good. But after some time, the fertility of the soil is decreases, and the again it will be problem for to grow the crops. So with this problem, we have thought about that, and we come to in means we started our working on this particular how to combat with this particular problems with the stresses, and particularly when we are talking about the biotic stress, how we will uh, combat with this problem. And in this particularly, we know that the plants microbes having a very good interaction, uh, and you know very well the microbes are present in the vicinity of the rhizosphere. So that's why it is called rhizospheric microbe. And but and also the microbes present in the plant inside. I mean, they have invade the plant and enter into the plant, and they uh, present there when we will take it out, when we will cut the plant stem or a leaf, and when we want to grow them, so that will be coming as an with the microorganism grow very well. And some of the microorganism, not all the culturable, some unculturable microbes are also present, which we cannot grow in on our patty plate in the lab, but we can grow some of microorganisms which grow that are culturable, and we can see that these culturable microorganisms are more sorry they are more having importance in combating with these stress conditions. So in that case, the and those who are microorganisms present in the plant, they are called endophytes, and they are having the some mutualistic and symbiotic relationship with the host plant, because the plants provide nutrition to the endophyte microorganism which is residing inside, and also plant get some immunity from that particular microorganism. So we can say that like in a human being, that particular microorganism which is giving the immunity to the plant to fight against this stress condition, whether it is a biotic or abiotic stresses. And of course, there's some microorganism which is are producing secondary metabolite, which we are very helpful in giving the good growth and other materials to the plant for enhancement for the production of the crop or the fighting against these stresses. As we know that the stresses, when we talk about the stresses, there are some external conditions that adversely affect the growth, development, and productivity of stresses triggers a wide range of plant responses. As already our previous speakers have given little insight on that, how this plant can respond. There are some plant that alter the gene expression, how to this combat with the alteration of the gene expression. Some cellular metabolism, they can change some growth rate and crop yield. So with these stresses, the biotic and abiotic stresses, there are some abiotic stresses like in a waterlogging, drought, high and low temperature, excessive soil salinity, inadequate mineral in the soil, and too much or too little light is also there are some abiotic stresses. And as already our previous speakers told about in the light, about the too much light and too little light about that one, how the plants behave in these particular conditions. So how this plant, as we, because dramatically I have tried to explain you the some environmental stresses. There are some environmental stresses. The severity, if the severity is there, because it depends on the severity of the stress, it is more means if it is a high severe stress, then how the plant will behave. If it is less severe, then how the plant is behave, and number of the some it depends on the stress condition, and in the stress conditions, all the plant tissues will behave stages and different kind of stage development of the plant, and some genotypes also means in the effect of the genes also there. If it is severe, so and if it's not much severe, so the plant behave into the response of the plant is of two types. One is the resistance, and one another is the susceptibility. So resistance means is survival and the growth of the plant take place, and when it is susceptible, so the death of the plant is take place. So with this stress resistance mechanism means the plant avoidance of the mechanism prevent exposure to the stress, tolerance mechanism permit to the plant to withstand the stress conditions, and acclimation means alter their physiology in response condition. With this means the plant itself under these stress conditions, they behave in such a manner and try to resist with this stress condition, and they make some changes either in the gene or in their molecular level or in cellular level and other in some nutrient level, so that plant can try to survive under these stress conditions. Now, how this changes in gene expression is response to the stresses. A stress response is initiated when plant recognizes stress at a cellular level. Stress recognition activities, signal transaction is there, pathways that transmit information within the individual cells and throughout the plant, and changes in gene expression may modify growth and development and even influence the reproductive capabilities. With this diagram, just this is, you can see with that, there are some distresses is there, and the behavior with this stress recognition is means first the recognition is the must. The plant recognizes the stress, whether the stress is severe or not very severe, and then after that it sends the signals to the cells, and then behave accordingly so that to combat with this particular stress condition. Now, I am talking about this flooding. When the crop is during the, because last year the Madhya Pradesh and whole India is having very heavy rains, and throughout the, you can say, season, lot of rains are there. So due to that crops, the flood situation is faced by the crop, and under these conditions we have done some experiments on the crop, particularly on the soybean crop system. So I will show you some photographs of this, because we have done on the field particularly, and this particular the organisms we have isolated as an from the rhizospheric of the um, same crop, so that we are using that particular endophyte and rhizospheres, so that how that behave when we will apply it into the field. So these flooding conditions will create a decrease of oxygen availability to the plant roots. That ATP production is lowered. In the survival strategies, production of enzyme for sucrose, starch degradation, glycolysis, ethanol fermentation, and ethylene for a long-term accumulation response, stem elongation is there. So with this means condition, how the our rhizobacteria and these conditions will help in that particular flooding conditions. This is the one of the slide which how it will be, what how the ethylene production is there, so that the elongation of the stem is higher, so that we came to know that the ethylene production is much higher because of some stress conditions is faced by the crop. Now, point is now our main focus. I am telling about the mitigating of the flood tolerance in soybean crop system by Pseudomonas, a potent PGPR microorganism. As this soybean, I am taking soybean is an important oil seed crop, particularly of the Madhya Pradesh. It is a cash crop of the Madhya Pradesh and some of other states also. And the rainfall is one of the most significant factors required for the proper growth and development of the crop plant, particularly in developing countries. And excessive amount of rainfall has its impact in the form of flooding, which has severe impact on the growth of the crop plant during this rainy season. Because some during the rainy season, and when the too much rains is there, the crop is facing the effect of these the pathogens attacks the crops, and due to the, because in the humidity is too much, as we know that when the humidity is too much, the growth of the fungus is too much. As we know in our houses also, when you see our walls, that particularly when during the rainy season, when the wall is moist, you can get the aspergillus or mucus like this fungus. So definitely when the humid condition is there, the chances of fungal pathogens, particularly fungal pathogens, is too high to attack the crops and other parts of the cropping system. So in this particular case, this is our field experiment. We have planned it, so that is the different way of that. Means we are taking the how much amount of we, because we have used this particularly PGPR microorganism, plant growth promoting microorganism, we have used in a different way, the different 100% biofertilizer, 75 to how to reduce the chemical fertilizer, particularly, and also the fungicide to reduce the amount of fungicide while using our this PGPR. So we came to know that we have also observed that particularly these when we use the biofertilizer, we are in a better way that we can reduce the quantity of means fungicide, and also we can reduce the use of a chemical fertilizer. So this is our I'm going to show you this particular slide that this, you can see that this is our treatment. We have uh, used this treatment. This is a soybean crop. And one thing I would like to say that our previous speaker, Dr. Sourav, very well pointed out, and one of that audience also they asked the about the germination of the seed, and we definitely I am agree with it point of Dr. Saurabh that there are a number of factors which are responsible for the seed germination. But I would like to say one thing in this particular case: when we use this particular plant growth promoting pseudomonas organism, and we will serialization this with this particular organism. So the rate of germination is increases by 10 to 20 folds. So when we use this particular, so if the, some seeds are means they don't want to germinate, but when we seederize the seed with this particular our organism, so the seed, the germination rate is about 90 to 98% is there. Now you can see this in this next slide. This is the slide where this the flood situation is there. The upper one is the flooded soybean variety. We have used it, and that is JS9560. And secondly, that is also a flooded soybean variety NRC7. So in that case, you can see this. This is the different variety which we have used. This is same variety 9560, and that is you can see the yellowishness of the particularly in this lower one. That is the B part. You can see the lowering means that yellowishness of the leaves is taken place, and in upper one that will be the treated one is there, that is the control one. So you can see in the control there is no treatment, so you can see the yellowishness, but in the treated field you can see you can see the greenishness of the leaves. So that is another slide which I would like to show you, that this is the control one, and in that particular control one you can see the plant situation is that there is no much leaves, no vegetative growth is there, and yellowishness is there. So this is that one, and we are when we compare with the control system, you can see that that the T1 is the control, and that is the R3 and R2. That is the treated with the fungicide and PGPR, and the T3. This is T2 is the this one treated one. Then again is T3. You can see that that particularly the T when we compare with the control system, you can see the vegetative growth of the soybean plant, which is very healthy growth is there, and you can see the color of the leaves is very good, and in that case the T4 is also is good, and in the T5 also you can see compare, and also you can see that when we will see the T4 and T3, particularly when you can see the T3, that there will be no attack of the you can say the insect attack you cannot see on the leaf. But in the case of the T4 also there are some attack you can see on the leaves, and T5 you can see the attack of some in the lower part of the attack of the insect is there. But in the case of the T6, that is very good. T6 is much more having impact, that is more greenish and also a good growth also. So with these conditions, that when we will see that some parameters like seed germination, as I already say that leaf chlorophyll and foliar damage score and oil content and grain, you can see that it is better in the T5 and also in the T6 you can see much more, and the green and that T3 also a very good comparison to the different other parameters. That is in the particular this table, the T3 is showing the seed weight per plant. We have taken that. So that is the T in comparison to the control. That is the T1 is the 3.62 grams, and in T3 13.64, and also you can see a 1,000 grain weight, because in the agriculture system we will take it out the 1,000 grain weight, because we will compare where there how much weight is there. So 1,000 grain weight will take it out. So in comparison to the T1, that is the T3 is much more having the impact on the crop production is much higher than the in comparison to the control system. This is another organism which is used as a Teluromyces trichosphermus. This is a fungus. Previous one was the Pseudomonas, another. This is and the Teluromyces trichosphermus. So we have also again we use and this particularly on the flood species, and we can see that this is the condition about the growth of soybean plant in control T1, left one and T2. This is right. T1 seed only treated with the talcum powder, 100% inorganic fertilizer. We have not used the in T2. We have not used the any means particularly that Teluromyces trichosphermus. Teluromyces trichosphermus, and in this particularly T2, we have used the RDF, recommended dose of fertilizer. Now, in the another case, you can see that the seed only treated with the talcum powder. The T1 is the same, but in this T3, you can see the 100% organic fertilizer we have used. No treatment of the our organism. Now here you can see that the T4, this is 100% Teluromyces. But here we can use, we have used only the 100% Teluromyces, our fungus. No other means we can say the treatment. No fertilizer, nothing else. Fungal, that is only the fungus we have used. You can see the condition of the crop. Here also in the T6 is there a Teluromyces plus 75% organic fertilizer we have used in T6. Now we see that the T6 after means when the flowering stage and other things, that is the T6, and you can see the growth of the plant. We have taken this photograph after the flowering, and this particular T6 is the Teluromyces and organic fertilizer. And here you can see the after 60 days when the pod of the T6 Teluromyces plus organic fertilizer, you can see the pod number of the pods and the number of you can see much more higher than in control comparison with the control. So this is the one of the table which can you see that the particularly the T6 is much more higher than in comparison to the T1. The germination rate of the percentage, as you can see, it is much more higher than in comparison to the control also. Now, this is the plant height we have taken. We can see in the T6 is much higher than in other parameters. Plant height with the after the sowing, you can see the 20 days, 40 days, 60 and 80 days and harvest stage. So the T6 is showing a good result in this particular case. Number of the branches, you can see the T6 having the much more branches in comparison to the other control and other treatments. Now about the pod number of the soybeans, also you can see in T6, T4, T7 and less you can find in T1 but is control and T2 and T3. Now, when we take out the grain yield per kg of plot, that is there P6 in 9.96, that is the mean, and total 29.87 in comparison to the control, that is 18.87. So, in comparison, when we compare the straw yield, that is much higher in the T6 because the growth of the plant is vegetative growth of the plant is very much higher in comparison to the control one. We have taken out the bundle yield per kg. You can see that it is also is much more in T6 and T4 in comparison to the other. So with this point, I would like to conclude and to say that when we will use the biofertilizer and of course the chemical fertilizer initially we have to use it, but with the chemical fertilizer when we are going to use the biofertilizer, and the farmers still they are not using much biofertilizer because they when we use the biofertilizer uh, the results which we farmers want to get it in a means in a faster way that the biofertilizer cannot give in a very much means faster way. So that's why the farmer they are using much more chemical fertilizer. But sometimes again they have to shift to the biofertilizer so that the fertility of the soil will be remain intact and the attack of the pathogenic microorganism and insect becomes less. When we use the chemical fertilizer, again we have to be use the uh, pesticide or insecticide to the uh, insecticide so that to kill the microorganisms. And, I don't know what happened. I'm coming or not? Hello, I'm audible. Okay.
Yes.
So particularly that I will conclude with this particular that when we use the biofertilizer, it will be more effective against to you can say the environmental stresses and also the insect means biotic and abiotic stresses, so that biofertilizer, particularly the microorganism, can fight against these stresses. And by listening to the previous speaker, Dr. Sourav, so that I have, because though I have 3 years of to work, 3 years I'm going to be retired, but still I would like to, because by listening his lecture, so that I came to know that still there are a lot of work we can do how this microbes can be reduce or can be, because the light, particularly when he has talked about the light, how this microbes can help in the about your HY5 like that, so that we can if the can we do it so together, that will be more helpful, because definitely microbes can also help the plant in make increasing the gene expression of HY5 like that. So with these words, I'm thankful to the organizers for inviting me and listening to the audience for hearing me. This. Thank you very much.
Thank you, sir.
Okay.
Thank you for such enlightening talk. Features of each cell and can promote the plant growth. There are few questions from the participants, which I am going to take. The first question is, apart from Pseudomonas and Saccharomyces, what are the other microorganisms which can be used for plant growth promotion?
Well, means apart from this, there are a number of microorganisms which we can use. The Bacillus species, there are a number of Bacillus species are we can use it, and other microorganisms we can use. You can look into the literatures which we can you can see that how these microorganisms, a number of different microorganisms can be used for this particular as a biofertilizer.
Question, sir. The process of action, both regulators, of action of how they are promoting, how they are helping to promote the plant growth.
It is a very good question, but it is very not well known how this fungus regulate the mechanism, because it is in everything, it is in the nature, it is they are doing. So it is very difficult to understand when we will do it in the our artificial environment. So the role of the microorganism, it is very not well known how it is they are behaving, because the microbes and plant interaction is still very I can say it is a very burning field to work on this particular line of action, because still a lot of work is required to know about the how the microbes work with the plant, so that this is interaction is very important to know about how they are working in the nature. It is very difficult to understand this.
So there are large number of questions. So
I think it is better you just circulate the my email to the participant, so that whatever they are having queries, they just share with me this, and I will reply them.
Say it something in the chat box also.
Okay.
One last question. Plant growth promoting microbes can be used singly or they better work in consortium? Veena Thakur, one of the participants.
Okay. Depends on what type of work you want to do it, because individual microorganism is working. It is more, I can say, if you are working, is more effective. But if you are going to on a multiple task system, like means you want to protect the plant with the stress, if you want to protect the plant with the pesticide, means pest, or if you want to protect the plant with other things. So on that case, on that particular point, I suggest that you have to use the consortium, because not an individual microorganism produce number of different kind of secondary metabolites. So, but to see that if the soil is not very rich, having the phosphate or whatever the nutrient, if you want to enrich with that particular kind of thing, a single microorganism, so that the plant can be enriched by that particular nutrient. I hope.
Yes, yes, sir. So one more question is there.
Okay, no worries.
This fungus which you have explained here, Saccharomyces, that is a broad range fungus that can be applied to number of pulses, or it is specific for any particular plant.
Okay. So this actually we have tried in soybean crop, and we have also tried in the wheat also. So that particular this fungus is working on both the crops very well. And in few means after 1 year, I hope that I will be in a position to the farmers to use this particular organism.
Thank you, sir. Thanks a lot.
Thank you
For this wonderful and informative talk. Now we have reached towards the end of today's session. And to propose a vote of thanks, I would like to invite my fellow colleague and member of organizing committee, Dr. Dhanushree Gupta. Over to you, Dr. Dhanushree. Dr. Dhanushree? Dr. Dhanushree, hear me? Dr. Dhanushree? Are you there? Audible? Excuse me?
Dr. Bhanusri ma'am, we are not able to hear your voice. Yes, ma'am, it is audible now.
Okay. Okay, so I'll start. Thank you, Dr. Joshi. On behalf of the organizing committee, I would like to express our heartfelt gratitude to our Honorable Vice Chancellor, Professor Keshrilal Verma sir, for his kind motivation and support to organize this event. We are also grateful to Registrar Professor Girish Kant Pandey for all the administrative and logistic support. I express our sincere thanks to Professor Kallol Kumar Ghosh, Director of Center for Basic Sciences, for his constant motivation, extraordinary planning, and overall supervision. We are extremely grateful to our esteemed speakers for making the event possible. Dr. Sourav Datta, Department of Biological Sciences, IISER Bhopal, has delivered a very informative talk on overview of plants, environmental stress. Professor Anil Prakash, Department of Microbiology, Barkatullah University, Bhopal, for sharing his knowledge on mitigating the impact of stress on plant productivity using plant growth promoting microbes. We do think that the lectures delivered today have certainly given a deep insight of botanical sciences. Our special thanks to print and electronic media for the wide coverage of the webinar. And in the end, I would like to thank all the participants, faculties and students, precious time and maintained with session. The webinar will definitely pave a path for new ideas, helpful in creating a knowledge base in botanical sciences, biological sciences especially. So we will meet you again tomorrow with the 3 important lectures by 3 eminent scientists. So join with us tomorrow at 11 a.m. Thank you. Over to you, Dr. Joshi.
Thank you, Dr. Bhanusri. So once again, I thank you all for joining me. And tomorrow evening, we start the session at 11 a.m. And the link for the feedback form for the both days will be provided and distributed in tomorrow's session. So now I am going to ask the motion in taking.